Welcome back to the course Fundamentals of General Immunology, and today we will discuss something related to molecular sensors of infection and cellular stresses or cellular stress factors. So what we have understood till now is when there is an infection, there is a huge amount of molecules which are entering inside the body. These are considered as non-self, and collectively these are called antigens. So in order to fight with those antigens or to identify those antigen, body cellular system is also having certain kind of sensors which are present on the either on the molecular surface or the cellular surface or on the internal cytoplasmic contents. So we will try to understand the details of those molecular sensors which are identifying those infection or stress granules post entry inside the human body. So when we classify these molecular sensors are classified as cell associated and the soluble one. So suppose this is a cell and we have this nucleus and this is cytoplasm. So the proteins could be, or these sensors could be present on the cellular surface, or they might be present sometime on the cytoplasmic area, such as here. They may be free floating or may be associated with some structures such as mitochondria, outer surface of mitochondria, or there could be a possibility that they may present inside a specific structure called endosomes. So endosomes are specific cellular compartments which are present inside the cytoplasm, and they are also having certain kind of sensors which can sense this antigen. Now we will discuss this in details later on when we will study the virus infection, but for time being we could understand that when there is a virus infection, these are internalized inside the host cell by receptor mediated endocytosis, and when they are endocytosed inside the body cellular system, then these are engulfed in a specific compartment called endosomes. So these are identified by the sensors which are present on endosome as well. So these are characteristic feature of some of the viruses. So what we are trying to understand that this could be a cell associated which might be present on the surface. The examples are toll-like receptors, nod-like receptor, RIG-like receptor. RIG stands for retinoic acid inducible gene 1-like receptor. The other could be C-type lactine-like receptors. There are scavenger receptors and also N-formyl methionine leucine phenylalanine receptors. So these are some of the things which are present on the cell surface. So these are associated structure. There could be possibility that some of the molecules can be solubilized outside and can do an effect. So once the cells are activated, the activated cells, so suppose these are activated cells, may produce certain kind of proteins, and these proteins are what you call soluble sensors such as pentraxins, collectins, ficolins, complement and natural antibodies. So these we will see gradually when we go along with this details about all these sensors. So let's try to do one by one, and we will try to finish a few of them in this lecture. So we'll start with the toll-like receptor. So this toll-like receptor is a very specific protein. This was discovered very initially in Drosophila as a part of innate immune sensors. So these are innate immune sensors. These are constantly present and very conserved evolutionary. So the toll-like receptors are the proteins which are either present on the smaller invertebrates to a higher vertebrate. So these are highly conserved evolutionary. That is what is mean by toll-like receptor evolutionary conservation. So this is actually first line of defense against the entry any pathogen that is entering inside the body. Few of them, as I told earlier, that there could be a possibility that they can present on the cellular surface, such as TLR2 and TLR4. So we could name the TLR based on the numbers, such as TLR1, 2, 3, 4, like that. So TLR2 and 4 are specifically present on the cellular surface, while TLR3, 7 and 9, they are present on endosomes, so the endosomal compartment. They can also be present on different cell types, such as macrophages and dendritic cell, which are known to present the antigen. So basically they are highly expressed on antigen presenting cells. So APCs are specific cell which can present the antigens, which we have discussed in the last class, to a specific immune cells, such as T cell and B cell population. So let's try to understand the receptor, its primary location, and the key ligands which they can identify. So TLR1, 2 and 6, sometimes they are heterodimeric structure. That means suppose if there is a surface, this protein can be either represented in the heterodimeric form. So heterodimeric form stands for the TLR1 could bind to TLR2 and make it as a heterodimeric protein. So therefore, you see that the surface can express multiple TLRs on the cell surface. So TLR1, 2 and 6 are located on surface, and they can identify the lipid or the lipopeptides which are present on the bacterial cell surface, cell wall content. TLR3 is very specifically present on endosomes. So we can see that TLR3, 7, 8 and 9, these are the one which are present on endosomes, rest all present on the cellular surface. So TLR3 is very specifically identifying the double-stranded RNA in the viral genome. We'll see what do you mean by double-stranded RNA, because even though there are viruses which are single-stranded RNA in nature, once they go inside the host cell, they can make another complementary copy of the RNA in order to replicate successfully inside the host cell. So there is a constant presence of double-stranded RNA molecules in the cytoplasm, which can evolve or activate toll-like receptor 3 sensing. TLR4 is again present on the surface, and these can identify lipopolysaccharide LPS on gram-negative bacterial cell wall. So these are very specifically LPS, what we call in the short, a very general term in the bacterial cell wall. So these are identified by toll-like receptor 4. Toll-like receptor 5 is again very unique. These are also present on cell surface, and they can identify the flagellar protein, the flagellin, which are present on flagella, and the bacterial flagella is helping bacteria to do a movement. And then toll-like receptor 7 and 8 is present on endosome, which can identify single-stranded RNA, that's again from the viral side. So the viral genome is actually comprising of single-stranded RNA or single-stranded DNA. So these are very specifically the sensor of single-stranded RNA moieties. Toll-like receptor 9 is again identifying CpG island. These are the specific DNA sequence which are rich in cytosine and guanosine residue. It's present in bacterial as well as the viral genome. And then there is very less information regarding TLR9, that their location, people are speculating that might be present on the cell surface and identifying some of the viral structures in order to sense that and raise an immune response towards the innate immune things. So these are some of the location TLR receptors and their key ligands. All right, we'll move forward and try to understand the toll-like receptor structure. So we discussed the toll-like receptor types and function and also location. Here we will look into the structure of TLR. So these are actually a cartoon of a protein structure, and we could identify that these are TLR1 and 2 heterodimer. Heterodimer means there are two different TLRs on the cell surface. So these are cell membrane, these are the hydrophilic moieties, and these are the hydrophobic, right? So they can hate water, so they can repel water molecules, and they can love water molecules. right? And therefore, these are decorated on the cell surface, and these are called transmembrane domain, and these are present on a very specifically the transmembrane protein, the protein which can span through the membrane. So, these are rich, since these are hydrophobic region. So, the protein is also mostly hydrophobic in nature. They tends to be buried inside. And then we have this cytoplasmic domain, which can be responsible for doing the downstream signaling biology. These are called TOL, IL-1 receptor domain, TIR domain. So, TOL is toll-like receptor and IL is interleukins. So, these are specific chemicals which are secreted between two leukocytes, and therefore, these are called interleukin. Interleukin means in between two lymphocytes or leukocytes. So, these are specific chemicals. So, TOL IL-1 receptor domains, TIR domain, is modulating the activity of this toll-like receptor through the cytoplasmic tail region. And then we have this area which is exposed outside that can bind to specific ligand in this docking area. These are the binding pockets here, and these are very rich in leucine amino acid. So, these are called leucine rich repeats. So, they are repeats of the leucine residue. So, sometimes there are 16 to 18 leucine rich repeats which are present here. These are highly again conformational, and they can allow the entry of, or say sensing of specific molecules which are identified for a particular toll-like receptor. So, this is a general protein structure of toll-like receptors. We will move to the next one, and next one is called nod-like receptor. Nod-like receptor is again a sensor which can activate or regulate inflammation. So, they can regulate inflammation, activate inflammasomes, which we have discussed in the last class, by this NLRP3, and trigger a very unique phenomena called pyroptosis. We have discussed about necrosis, we have discussed about apoptosis. This is specifically pyroptosis. This is also a programmed cell death like apoptosis, but the phenomena of activation of pyroptosis is slightly different as compared to apoptosis. So, we will see in detail a little later. This NLR are proteins again very rich in leucine repeats, similar like toll-like receptors. So, these are the area which can be sensed by specific molecules or antigens. Then we have this NACST domain, which is a kind of a mixture of 4 proteins which are listed here. I am not going to discuss those names, but one can always look into this. So, these are kind of domains which are conserved. So, this is NACST domain which is in the middle, and then finally, we have this pyrene domain which is there in the cytoplasmic tail, cytoplasmic area. Now, this specific pyrene domain can activate the downstream caspase related pathways also. This can sense and then pass on information through the pyrene domain. So, this is a nod-like receptor which can trigger the process of inflammasome formation. So, since we started this topic of pyroptosis, let us try to understand this phenomena of pyroptosis. So, the critical thing which we need to understand here in pyroptosis is caspase 1, the middle one. So, the caspase, as we discussed, that these are very important proteins which are cleaving, these are cysteine aspartate proteases which are known to modulate multiple pathways. So, there are caspases which are involved in apoptosis like 3 and 7. Here we will concentrate our discussion to caspase 1, which is involved in this pyroptosis phenomena. So, this caspase can activate multiple pathways. So, we can just see one by one. So, this caspase, activated caspase, can do cell swelling and lysis. So, accordingly, we can see that there is a lysis of the cell. This can also cause the independent pore formation, which can allow the entry of ions or exit of the ions, also the entry of water molecules. So, these are making the holes in the membrane sometime. They can also make a hole in order to do some secretions from the cellular system. So, here you can see all the ions or water is going out and in. Here there could be a possibility that some of the cellular content can go also out. They can also cause the cleavage of bigger cytokine into an active form. So, the cytokine processing could happen because of the caspase-1, because these are proteases. So, they can cleave the pro form of this cytokine, such as pro IL-1 beta, which we have looked into the last class, can be converted by cleavage into IL-1 beta. So, this can be do and trigger the inflammatory processes. So, they can either go by this microvesicle shedding, which can happen in the cellular membrane, or they can also do a lysosome exocytosis, where the things can be degraded by lysosome and then do an exocytosis. So, this can do multiple pathways related to this, ultimately leading to the cell damage, and this can lead to the cell death. And all these phenomena, since these are governed by caspase-1, these are called pyroptosis. So, one can easily say that any phenomena that is controlled by caspase-1 is what we call pyroptosis. They can also have an endonuclease mediated DNA cleavage that can happen with caspase, that could lead to nuclear condensation. So, one can understand that there could be multiple ways caspase can do cell death, and that is what we call collectively as pyroptosis. So, this is just one term which we need to understand when we discuss about the nod-like receptor. Alright, we will move further and we will try to summarize this TLR and nod-like receptor in terms of gram-positive and gram-negative bacteria. So, we know that gram-positive, if we stain with a gram stain, we can see violet stain color, and gram-negative is what will give a saffron in red coloration upon staining. We have just tried to draw here that these are some of the flagella which are present in the gram-negative bacteria. So, we have DNA, lipoprotein, peptidoglycan and lipoteichoic acid, which are present in gram-positive bacteria, and they can be identified by lot of sensors, such as the cell surface receptor can identify lipopolysaccharide, peptidoglycan, and also cell surface receptor can identify the lipopeptides, peptidoglycans, and also lipoteichoic acid, which are present on gram-positive bacteria. While here the LPS, lipopolysaccharide, and peptidoglycan, these are identified by a set of TLRs, while specifically if there is a flagella, these are identified by TLR5. So, we are just trying to highlight and recapitulate the discussion which we have done with respect to the TLR and nod-like receptor, and then we have the intracellular receptors such as CpG specific DNA molecules can be identified by TLR9. Nod-like 2 receptor can identify the peptidoglycan, then again the same goes here in gram-negative bacteria as well. So, this is just a pictorial representation of the different sensors which are present to identify the gram-positive and gram-negative bacterial antigenic contents. Right, we will move on to the third one, which is called RIG-like receptor. RIG stands for retinoic acid inducible gene 1, which is again a gene that get activated by vitamin A. Retinoic acid is vitamin A. Then we have this second one called MDA5, which is a myelinoma differentiating associated gene specific protein, MDA5. So, these are intracellular sensor again and identify non-self viral RNA. So, we could look into that why this viral RNA. So, there are RNA which are present in the host cell also, which can form the proteins of post binding to the specific ribosomes, and again we see that suppose you have a cell, the RNA which are coming out from the nucleus post transcription can bind to the ribosome to form a protein. Now these RNA are self in nature, but when there is an infection of a virus, the viral carrying the RNA can also come in the cytoplasm. So there could be a sensors present in the cell that can identify this as a non-self and destroy, and this identify as self to promote the translation. And therefore these are the sensors which we are discussing here, called RIG-like receptor. So these are specifically present in context to the cytoplasmic area, and therefore these are cytoplasmic sensors. And one can see that they have a C-terminal domain, they have a central helicase domain, and then they have a caspase active recruitment domains, two of those domains that can recruit the caspase protein on the cytoplasmic tail to activate it. So these are the two card-like domains which are present on their other side of the protein. So they have an helicase domain which can actively do helicase activity by utilizing an ATP. So this is an active process, and then C-terminal domain can link with the specific RNA that's coming from outside, and most of the time this is a viral specific RNA, which we will see in the next slide. And these are the CAD domain which is present in the cytoplasmic side to activate this pathway. So these are activator of this whole process. So let's move on and see that how these are happening. So these are, as discussed, these are the cytosolic sensors. So once there is a virus infection, the virus is coming into the cytoplasm and doing this single stranded RNA throwing inside the cytoplasm. They have a 5' phosphate residue sometime added to the genome. The examples are influenza viruses, paramyxoviruses, and flaviviruses. Flaviviruses are such as dengue, Japanese encephalitis viruses, which are carrying RNA. So the difference here is the influenza is a negative sense RNA carrying viruses. Paramyxoviruses is also negative sense RNA carrying viruses, while flavi is positive sense RNA virus, positive sense RNA carrying viruses. And of course there are other differences which are present, like influenza we see that the genome is segmented. So the genome is coming in multiple segments, sometimes the segments are 8 in number, and they are coded by a viral RNA polymerase into a protein differently. So these are segmented, while paramyxoviruses and flaviviruses are having a single RNA molecules in their genome. Now anyway, these single stranded RNAs are coming into the cytoplasm, and they can be sensed by this helicase domain which are there. So the card is in the fag end, and this is the helicase domain, and these are the front domain, the C-terminal domain, which can identify this RNA. Once they are identified, these become active and then do mitochondrial antiviral signaling protein got activated, and this can lead to the production of interferon related pathways. So essentially these are all antiviral signaling that can lead to antiviral effect in the host body. So these are related to single stranded RNA that can activate the RIG1 protein which are present in the cytoplasm. We could also understand that the viral RNA can sometimes form a double stranded RNA intermediate. Well, there are viruses which carry double stranded RNA such as rotavirus. So rotavirus or reoviruses are actually the viruses which carry double stranded RNA genome. While in context of this discussion, we will try to see that the double stranded RNA could be formed as an intermediate during the replication of the same single stranded RNA. So this double stranded RNA can also be sensed by another protein called MDF5, and this MDF5 is exactly the similar kind of domain. So they have this helicase domain and the card domain, and this RNA will be sensed by this C-terminal domain and then activate the same pathway. So these are actually two sensors which are look like a RIG-like receptor sensors. So RIG1 and MDF5. So these are the two very specific sensors which can detect viral RNA which are non-self in nature. Okay, we'll move to the next one called C-type lectin-like receptor, CLRs, and these are very specifically a receptor which can detect the carbohydrates and other ligands to initiate phagocytosis, inflammation, and shape adaptive immunity. So these are the receptor which can detect carbohydrate most of the times. There are two examples which we have listed here. One is called DECTIN-1, which it can detect the fungal cell wall antigens, and the DC sign, which are very specifically the sensors which are present on dendritic cell, and they can detect all the dendritic cell associated structure which can be processed through dendritic cell. So these are the sensors which are dendritic cell associated. These are actually defined by a specific, at least they have one C-type lectin-like domain, CTLD they used to call, which can bind to different ligands which is discussed like carbohydrates, which could be mediated with or without calcium. So this is again very important point. These are calcium dependent or independent pathways related proteins. The other important thing is these C-type lectin receptors are actually modulated through these ITAMs. These ITAMs are actually immunoreceptor tyrosine based activation motifs which are present on the cytoplasmic domain of a protein. So I'll just try to show here roughly, and then in the next slide we'll see the details. So suppose this is actually a lectin-like receptor, and then there is a cytoplasmic domain, and then in the cytoplasmic transmembrane, so there is a transmembrane domain. So let's try to understand these ITAMs. So these ITAMs are actually a specific receptors activator which are present on the cytoplasmic tail of a protein. So for example, we'll try to understand here in a specific cartoon, and then in the next slide we'll discuss the detail. So suppose this is a C-type lectin, and there is a transmembrane domain here, and then we have a cytoplasmic tail. Now this ITAM are specific immunoreceptor tyrosine based activation motifs which are activated post phosphorylation. So once the kinase attack here, so the kinases are the proteins which can add the phosphate here by utilizing the adenosine triphosphate and then convert it into adenosine diphosphate. So the phosphate can be added here, and this protein is now become active. So to activate this protein, the phosphorylation of this ITAM is important. So many of the proteins which are immunoreceptor in nature must have this ITAM motif on their cytoplasmic tail, and this, for example, here in C-type lectin, the carbohydrate moieties will be identified here, and they can, once identified, then they can activate through this ITAM receptor. So this is just a signaling cascade of this C-type lectin. So let's try to understand this in a detailed form. So here we have this fungi or yeast, which is again a eukaryotic cell. So they have lot of details on their surface, and some of the details are actually the complex polysaccharide which are present on the cell wall of fungi, and this can be identified by this lectin receptor called DECTIN-1. So they can be identified with this DECTIN, and then we have this transmembrane domain here which can be terminated into this cytoplasmic domain. So this is an ITAM residue here, and one can look into that. There are kinases such as here, for example, SYK, which is called spleen tyrosine kinases, and these spleen tyrosine kinases can phosphorylate this ITAM, which can activate the downstream cascade, which can activate NF-kappa B to do a downstream inflammatory signaling responses. So the binding could lead to phosphorylation of these cytoplasmic protein, which can activate this whole process to do a cytokine production, which could ultimately lead to the raising of the immune response, either in the form of adaptive or any other way, such as inflammatory responses, in order to nullify this infection which is coming from outside. So these are all some of the receptors which are listed here. We will go in detail about the other receptor in the next class. Thank you.